flavonoid is a major class of phytochemicals that is widely found in plants. Flavonoids are polyphenols, and increasing evidences have shown that flavonoid intake can be beneficial to health. Flavonoids have several health-promoting properties, and studies have shown that flavonoids consumption can have positive impacts on cardiovascular health. Epidemiological studies have reported an inverse correlation between the intake of dietary flavonoids and the mortality rate from cardiovascular diseases. Studies have been carried out to investigate the effects of flavonoids on the cardiovascular system. In many of these studies, flow-mediated dilation, also known as FMD, was used as the research tool to predict the future cardiovascular events. FMD is a non-invasive research method in evaluating the endothelial function. FMD is also a functional surrogate biomarker for the risk of cardiovascular disease. FMD is the measure of the increase in vascular diameter, followed by a transient ischemia. A low level of FMD is associated with an increased risk of developing hypertension, chest pain, and congestive heart failure. The regulation of the blood vessel diameter in response to physiological needs is mediated by several key enzymes, such as prostacyclin synthase and endothelial nitric oxide synthase, also known as the eNOS enzyme. eNOS is the key enzyme that catalyzes the conversion of L-arginine to nitric oxide. Nitric oxide is the central molecule involved in the modulation of vascular tone. In fact, it has been suggested that the bioactivity of nitric oxide provides a functional readout for FMD. Nitric oxide is a vasoactive molecule. Vasoactive molecules are endogenous compounds or pharmacological agents that can regulate the heart rate by adjusting the vascular resistance. The regulation of vascular resistance by vasoactive molecules is through the process of vasoconstriction and vasodilation of the blood vessels, and the regulation of vessel diameter in response to physiological needs is the key of maintaining the body's homeostatic mechanisms. Nitric oxide, being a vasoactive compound, acts as a diffusible mediator in the smooth muscle and regulate the vessel diameter. Interestingly, studies carried out on human volunteers have shown that the consumption of flavonoids-rich beverages led to an increase in FMD, suggesting that flavonoids can have positive impact on cardiovascular health. Further studies carried out to understand the mechanism of action of flavonoids have revealed that flavonoids can affect the FMD levels in two ways, which include acute and chronic effects. Studies have shown that the elevation of FMD level was observed a few hours after the consumption of a single intake of flavonoids-rich beverage in human volunteers. This instant physiological response to flavonoids suggests that the molecular mechanisms that underlie the FMD response are most likely through the post-translational modulation and regulation of pre-existing signaling cascades, rather than the regulation of gene expressions and protein synthesis. It is very likely that flavonoids act by the activation or inhibition of enzymes and molecules that are involved in the nitric oxide synthesis pathway, which in turn leads to an increased blood flow through the cardiovascular system. In fact, studies have provided evidences for the modulatory effects of flavonoids on the key enzyme that is involved in the nitric oxide synthesis pathway, which is the eNOS molecule. It is suggested that the consumption of flavonoids leads to the phosphorylation of eNOS at serine 1177 residue. The changes in phosphorylation pattern of eNOS promotes the catalytic activity of eNOS enzyme, which in turn leads to an increase in the nitric oxide bioactivity. Nitric oxide is a key mediator of vasodilation. Nitric oxide interacts with guanylate cyclase in the smooth muscle cells within the blood vessels. The activated guanylate cyclase converts GTP to cyclic GMP, which is a second messenger molecule. cGMP in turn activates the cyclic nucleotide-dependent protein kinase G, also known as the PKG molecule. The activated PKG molecule catalyzes the phosphorylation of molecules that are crucial in the regulation of calcium concentrations within the cell. The increase in calcium concentration within the cell leads to the efflux of potassium ions through the potassium channels. The hyperpolarization of smooth muscle cells of the blood vessels alters the dynamic of actin filament and myosin and results in the relaxation of smooth muscle cells, thereby resulting in vasodilation in the blood vessels. Furthermore, multiple studies have shown that flavonoid intake can have chronic impact on the FMD level. The chronic effect of flavonoids involves a chronic elevation of FMD level by sustaining the basal FMD level at a higher set point. It is suggested that the regulation of FMD set point is through the regulation of the transcription profiles of the enzymes within the FMD regulation cascade, such as eNOS, which is the key enzyme involved in the metabolism of nitric oxide. It is also suggested that flavonoids are able to regulate the transcription profiles of enzymes that are involved in the biosynthesis of arginine, which is the precursor of nitric oxide. One example of the enzymes that are involved in arginine biosynthesis is arginase. For example, Lehrer et al. Demonstrated that flavonoids exert its effects on eNOS through the upregulation of eNOS transcription factor CRAPA-like factor II expression, which in turn results in an increased depression of eNOS.